Good morning. My name is Pratibha Singh. I am assistant professor at Wells Center for Pediatric Research uh, at Indiana University of School of Medicine. Today, I will talk about the basics of flow cytometry and its application. I will talk about how a flow cytometry works, what are the flow cytometry components, how are flow cytometric data displayed, and what are the flow cytometry applications. As name suggests, flow cytometry measures the cell in a flow or in a stream. Flow cytometry is a laser-based tool that measures the biological and chemical properties of cells or particles flowing in a fluid in a in single cell stream. Flow cytometry is a powerful tool to measure the biological and chemical properties of the cells due to following qualities. It can do rapid analysis. It can measure 3,000 to 200,000 even per second. We can do individual cell analysis. Multiple parameters can be analyzed at same time. We can get quantitative data with statistical significance. The basic here I showed the basic mechanism of a flow cytometry when cells are labeled with fluorescent conjugated antibodies or reagents and inserted in a flow cytometry for data acquisition. The cells move in a single cell stream and intercepted by a laser beam. When the uh, fluorescent conjugated cells are interacted by the laser beams, fluorescence molecule gets activated and emits light of a higher wavelength. That light is filtered and detected by photomultiplier tubes, and the light and fluorescence are then converted into electrical signal and followed by in digital signals. This schematic shows the different components of flow cytometry. Flow cytometry measures the light scattered by the cells and the fluorescence emitted by the fluorescent level cells when they are interacted with the laser lights. So when fluorescent label cells are injected in flow cytometry for acquisition, so fluidic system it forces the cells to run in a single cell stream, and there the laser beam interact with the cells, and the cells after interacting with the laser beam, uh, cells scatter the light in two directions, one in the axis of the laser beam and then another in the at 90-degree axis of the laser beam. In addition, the fluorochrome molecules get activated and they emit the fluorescence, and this the fluorescence also scattered at the 90-degree angle of the laser beam. Then the optical system, it filters the different wavelengths of the emitted the fluorescence and then detected by the different detectors. Then the electronic system that convert the light and fluorescence into the electronic signals and followed by digital signals. Flow cytometry was made feasible due to three important discoveries. And first one is antibodies, the second one fluorochromes, and then lasers. Antibodies bind to a specific marker. It can bind to cell surface or intracellular. Although antibodies are important component of the immune system, but for flow cytometry, they are used as a tool to bind to a specific proteins that express on the cell surface or inside the cell. There are two types of staining can be performed to detect the specific markers. First one is direct staining, and the second one indirect staining. In direct staining, the cell surface markers are directly bind to the fluorochrome conjugated antibodies. Whereas in indirect staining system, cell surface markers are first bind to the purified or bioattenuated conjugate bioattenuated antibodies, and this purified or bioattenuated antibodies then bind by fluorochrome conjugated secondary antibodies. Fluorochromes are the molecules that absorb light of a specific wavelength after excitation with a laser beam, and then it re-emit the fluorescent light of a longer wavelength. There are several fluorochromes molecules are available from different vendors. Some examples are Ph.D, PE, PE size 7, Electra 4488, Pacific Blue 405. Lasers are used to activate the fluorochromes. Lasers provide light of a specific wavelength needed to activate fluorochromes. Each fluorochrome each fluorochrome is activated by a specific wavelength of light. So most of the flow cytometries are equipped with 2 to 4 lasers, and each laser can emit a specific wavelength of light. For example, laser 405 that emit the wavelength of light of 405 nanometer. The green laser that it is 488 that emits light of 488 wavelength. In the different fluorochromes, they absorb light of a specific wavelength range, and then emit light of a specific wavelength. Here is the example that Ph.D. It can be excited by 488 lasers, and although it can excite over a range of wavelength from 400 to 650, but the maximum absorption happens the at uh, at 550 wavelength, and the this fluorochrome, when it emits light after excitation, the maximum wavelength that can be emitted is at 650 nanometer wavelength range. Uh, so um, when cells or particles interact with the laser beam, the cells or particle can scatter light in all directions. But in flow cytometry, we measure light that are scattered in two angles or two directions. So one is forward scattered, and the second one is side scattered. If forward scattered is where the light scattered in the axis of the laser beam, whereas the side scatter is the light that scattered at the 90-degree angle to the laser beam. Light that scattered at in the axis of the laser beam, it measures the size of the cells, whereas the light that scattered at the 90-degree axis of the laser beam, it measures the granularity of the cells, or complexity of the cells. The fluorochromes are also fluorochromes, when they get excited and they emit the light of fluorescent light of a higher wavelength, that fluorescent light is also scattered at the 90-degree angle. So the size of a cell is determined by the forward scatter, whereas the complexity and the fluorescence of the cells are determined by the side scattered and the fluorescence that are the fluorescent light that is scattered at the 90-degree angle to the laser beam. So the light that is scattered at in a linear axis to the laser beam, and 90-degree linear axis to the laser beam, that measures the size of the cells, and the light that is scattered at the 90-degree axis of the laser beam, that measures the complexity of the cells. So together, the size scattered and the forward scattered, these two parameters measures the cell size and complexity. And the fluorescence light that emitted at 90-degree axis of the laser beam, it measures the cell surface markers expression on different cell types. A flow cytometry is made up of three main systems: fluidic system, optic system, and electronic system. Fluidic system, it transports cells or particles in a stream of fluid to the laser beam. Optics, it consists of lasers and optical filters. lasers activate the fluorochrome of label cells, and then optical filters direct the emitted fluorescent signal to the detectors. Electronics of a flow cytometry system, it converts the detected light signals into electronic signals that the computer can process for digital data. This picture shows the different component of a flow cytometry. So the fluidic system that force the cells to align in a single cell stream. The lasers that used to scatter the light by the cells or particles, and also used to activate the fluorescence label cells. And when the light is scattered in the linear axis and at the 90-degree axis, these light and fluorescence are detected by the detectors and filters of the optical system. And then the electronic system, it converts the light energy and fluorescence energy into electronic signals, and then in digital signals. The fluidic system of a flow cytometry works on hydrodynamic Fock system. So the fluid system is made up of a flow cell where the seed fluid, that is the outer seed that surrounds the cell suspension, that is in the center, and it force it to travel along the axis and pass through the laser at one cell at a time. The optical system has two parts, excitation optics and collection optics. So excitation optics, it comprises of lasers and lenses. Lasers and the focusing lenses, it produce a beam of light that when that interacts with the cells or particles at the fixed interrogation points and excite it. Whereas collection optics, it composed of filters and mirrors. The light scattered produced from the cell laser interaction is collected by filters and directed to either a photodiode that for the forward scatter, or redirected via a system of mirrors and filters to the appropriate photomultiplier tubes, and that is for the fluorescence and the complexity of the cells. Each laser, the optical system, is composed of multiple lasers. So the advantage of using multiple lasers are that it allows to increase the number of markers that can be examined at same time. Each laser emit light of a specific wavelength. So 4 or 5 laser that excite the fluorochromes at 405 nanometer wavelength. Whereas 488 lasers that excite the fluorochromes at 488 nanometer wavelength. Fluorochromes that are excited at different wavelengths can be used in a combination. So even if they emit similar wavelength of light, but but still, if they can be used together, and by using filters and detectors, the emitted wavelengths can be separated for the different markers analysis. So the collection filters, the function of the collection filters are that they separate the different wavelengths of emitted light. When when a fluorochrome conjugated or labeled cells are are interrogated by a laser beam that and they emit light of different wavelengths. So the different wavelength of light are simultaneously generated, and these light must be split into its specific wavelengths to measure and quantify them independently. And the optical filters, it can separate the different wavelengths of emitted light. Optical filters are designed in such a way that it can absorb or reflect some wavelength of light while transmit other wavelengths of light. So there are three main types of optical filters: long pass filters, short pass filters, band pass filters, and diachronic filters that are like another type of optical filters. So for long pass filter, so the long pass filter, it can transmit wavelengths of light above certain wavelength. So for example, long pass 500. What does it mean? That when the different wavelengths of light um, interact with this filter, so it transmit only the wavelengths that are above 500 nanometer wavelength. And short pass filter that transmit wavelengths above transmit wavelengths of light that are below certain wavelength. So short pass 500 filter, what it does, it when incoming lights wavelengths of light interact with this filter, it transmit wavelengths that are below 500 nanometer. And band pass filter, it transmit only a range of wavelengths. So example here that band pass 530 slash 30 nanometer filter. What does it mean? That it transmit wavelengths of light that are in range of 530 nanometer plus 15 and minus 15. So it can transmit. So it can transmit wavelengths between 515 to 545 nanometer range. This is an example that how a mm, different uh, optical filters can separate different wavelengths of light that are emitted by a fluorescent molecule. So, if this the these four fluorochromes FITC, PE, PERC, Psi 5.5, and PERC and PE Psi 7, they all are excited by 488 uh, mm, lasers. And when they, after excitation, when they emit fluorochromes, they emit different wavelengths of um, light, fluorescent light. So the different optical filters can separate the emitted light that are emitted from different fluorochromes. So, for example, uh, this 530 slash 30 uh, filter, optical filter, can uh, can filter the emitted fluorescence from the FITC fluorochrome. Uh, this 585 slash 42 filter can separate emitted light from PEA fluorochrome, and the similar way using these two filters uh, we can separate emitted fluorochrome emitted fluorescence uh, wavelength from PERC Psi 5.5 or PE Psi 5, PE Psi 7 fluorochromes. Once we have the uh, the the emitted light, fluorochrome light is separated in different based on different wavelength, then the electronic system converts the signals from the detectors uh, into digital signals that a computer can read. Once the light signals strike the photomultiplier tube or the photo diode, they are converted into a relative number of electrons that are multiplied to create the electrical current, and the electrical current moves to the amplifier and it is converted to a voltage pulse. And then ADC converts the pulse into a digital signal. Flow, there are several applications of flow cytometry, which include flow cytometry can be used to do phenotypic analysis of cells. We can identify different subpopulation of cells in a heterogeneous sample. We can measure the cells' biological properties, their cell proliferation, cycle, DNA content. Yeah. In addition, we can examine cells survival, their the autophagy. There are several properties that we can measure with flow cytometry. We can also measure cells' functional activities, including mitochondrial activity, gene expression, calcium flux. These are some example where flow cytometry can be used. Uh, there are several types of flow cytometry are available from different vendors, including traditional flow cytometer, which use mirrors and filters to capture the emitted wavelength from excited fluorochromes. Um, the fluorochromes, the limitation with this type of flow cytometry is that the fluorochromes that have emission profile that fall into the same filter cannot be separated from one another. The cell sorter. The cell sorter is also it falls in category of traditional flow cytometry, but it allows that to collect the samples after the processing. And the sorted cells we can use for different function analysis. Imaging cytometry. The imaging cytometers or traditional flow cytometer that combine with the fluorescent microscopy feature. An imaging cytometer can measure morphology and multiple parameter uh, of fluorescent cells, both in a single cell level and at population level. There's like another type of flow cytometer that is called CyTech, a spectral cytometer. Spectral cytometers capture the full spectral profile of the fluorochromes across all layers, and the fluorochromes profiles are captured in 10 to 30 nanometer segments across the emission range, and to accurately unimix the fluorochromes emission spectra. This the Because in this cytometer we can measure the whole spectral profile, so it increases the number of fluorochromes that we can measure at a time. We can measure up to 54 different markers at the same time. So in summary, flow cytometry analyzes multiple parameters of individual cells in a heterogeneous population that are flowing in a fluid stream. Flow cytometer uses the principle of light scattered property of a cell and excitation and emission uh, uh, properties of the fluorochromes to get the data from cells ranging from various small to big size cells such as 0.2 micron to 150 micron in size. Flow cytometry can be used in multiple applications, including phenotypic analysis, cell cycle, and survival. Here, I would like to stop. Thank you.